Okay, thanks, Kate. It's really great to see everybody. So thank you, first of all, for coming. I'm Ryan Kelly. I'm the director of the eDNA Collaborative. And this is, some of you heard as we were signing on, this is the third of these three meetups that we've done this week. And it's our first, this is the first time we've done this. This is the third of those, but before this week, we had never done it. And it's been really a great opportunity to sort of see people interact with each other and get to hear what folks are up to around the world and have them introduce each other. You introduce each other, to introduce yourselves to each other. Sorry, it's a bit late in the day here. It's only 6 o'clock, but still it feels like dinner time. And so it's actually really nice to have a little bit of a smaller group. We had more people earlier in the week, and yeah, just really very exciting. So let me give a just very brief introduction here that about 18 months ago, we started up the eDNA Collaborative with the idea of moving eDNA from theory into practice, into routine practice around the world. And we thought, well, how can we do that? What are different theories of change that we may make use of and avenues through which we might make eDNA, sort of normalize eDNA through the world and have it be more accessible to more people. And so we do that in these different ways, including, doing our own core research. We've been able to make a couple of pilot grants for small projects. We've had visiting scholars, 6 of them in total, come and visit us and hang out with us in Seattle and do a two-way exchange of information for about a week. But really, these microgrants have become more core to what we've been doing. It's been a great success to be able to give small grants in the form of equipment or services or whatever people need to try and help reduce barriers to entry to being able to use eDNA in your research. And so everybody here has at one time or another received a micro grant, and we've now given out more than 100 of these to people in over 40 countries. So it's felt like just a fabulous way to be able to know who's out there, know what they're doing, and try and help a little bit. And so the next step is this, where we're trying now to, if we know who each of you is, we now want you to know who each other is and stitch yourselves into a network of people so that you have friends to rely on. You have people you can ask questions to. And no matter what you're working on, we actually all have a lot in common in terms of the different techniques we're using and different tools. So let's get into it. We're going to have the rest of the team introduce themselves here on the next slide. Kara, take it away. But you're on mute.
Thank you. It's late, like Brian said, but hi everybody. It's nice to see you all on the screen. I know I've emailed with almost everybody here when you received your microgrant in order to coordinate. And I'm really excited to see your work and hear what you're doing with the microgrants and beyond. My background is I was an engineer and a geochemist. And then I ended up working in the logistics side of science support for about 20 years in the Antarctic. And now I've made my way over to the collaborative, which has been really exciting and really interesting. And I am working with both Eiley, Kate, and Ryan on helping coordinate these microgrants and other things that the collab is doing. And it's been really fun. Eiley.
Thanks, Kara. Hi, everybody. Nice to see you. I'm really excited to hear about your research and see your slides. My name is Eiley Allen, and I'm the chief scientist here at the collaborative. I've been doing eDNA work for about 10 years. I mostly study fish and marine environments, but have really broad interests and love hearing all of your diverse taxa and environments. So we're really excited to have you here. Thank you again for volunteering to give these slides and take this opportunity to meet each other. We're going to get right into the slides, and then I think Kate mentioned I'll be timing. So after about 3 minutes, I'll just put a little note in the chat if you're going a little long. And after I introduce the first person, you can introduce yourself when your slide comes up. But our first person is actually Antonio Beza, who does not have a slide. But Antonio, if you want to unmute yourself and give your little 2-minute talk on your research, Then we'll pick up with slides after you have finished. Thank you.
Oh, sounds good. Thank you so much, Elizabeth. Thanks, everybody. Apologies for not seeing the slides, sort of like a heavy semester already here. My name is Antonio Baeza. I'm originally from Chile. I have been for more than 10 years in the States. Right now, I'm an associate faculty at the Clemson University, that is South Carolina, East Coast, 9 p.m. right now here. Originally, I did a lot of behavioral ecology. I was interested in studying sexual systems in just one species. I was interested later in understanding if that sexual system evolves several times. So I got a little of knowledge on phylogenetics. From there, barcoding, and I retooled myself while a faculty at Clemson here with genomics. I have several interested in genomics, classic ones, phylogenomics, phylogeography with genome-wide markers, but in terms of environmental DNA, I'm very interested in trying to obtain whole mitochondrial genomes from environmental DNA in the form of scats. I'm doing what we call right now pupomics. Sorry for the bad joke, but pretty much it's trying to obtain as much genome as possible from different type of feces. We have been successful. We have a paper recently published in Romero Lagos Dayasi. That is a little small rabbit high in the mountains in Mexico, endemic species. Really, because they have conservation issues, we're able, maybe ideally, now that we know that we can assemble full mitochondrial genomes from droppings, yeah, we will try to use it as a tool for biomonitoring and bioprospecting on the one hand again. We have been successful with tigers, pandas, whales, and in the future, ideally, we will try to do phylogenomics or phylomitogenomics using, again, environmental DNA in the form of scats. Thank you so much again for the opportunity. Great, and appreciate the microgrant. And thank you. Next, I guess.
I don't see Dale Ann in the participants, so let's give one more second here. If you are here, let us know. Otherwise, we can come back to the Dale Ann slide at the end.
I also don't see Jessica, so we'll progress too.
I wonder, Kate, is there a chance these are the slides from the other day? Because we had we had these people yesterday.
Yeah, they some people signed up for multiple meetings. So Joyce, maybe
yeah, here we go.
Joyce.
Yeah.
Okay. Hola. Hola. My name is Joyce Gomez. I'm currently a master's student in oceanography at the University of Puerto Rico in Mayagüez, where I'm investigating the microbiome of corals affected by the stony coral tissue loss disease in La Parguera, Puerto Rico. In Puerto Rico, we have a lot of corals diversity, which have been impacted by different diseases. As some of you may remember, this disease was first seen in Florida in 2014 and quickly spread to the Caribbean and Puerto Rico. Although it impacts more than 22 species of corals, there are different variations of diseases which have not been studied enough to recognize the cause. We have, for example, the white band and the black band disease in Puerto Rico affecting the coral reefs. We thought it would be interesting to see if there are any differences in the composition of its microbiome before, during, and after the disease. Sadly, once the coral has been impacted, it loses its tissue and there is no mucus to extract. Therefore, these samples are not taken into consideration, but we are using the before and during the disease. For this project, I am working with different corals, but mainly with Orbicella fabiolata, Pseudoplorea strigosa, and Colpophilia natans. We do the less invasive sampling method. This is to scrape the coral for the mucus to levitate in water. Then we take a syringe and we take the mucus all together with that water. These samples are preserved in its own ranges at around negative 60 Celsius degrees in an ultra freezer. When needed, these samples are thawed and its DNA is extracted with the DNEC Power Soil Pro Kit using 16S as a tool to identify these bacteria. Then the next step is to do PCR and sequence these samples. We are currently on this last two steps of the project, doing the PCRs and sequences, but we have high hopes to see a difference in their microbiota before and during this disease. Some good future collaborations would be coral restoration on a molecular level or some new takes in coral's ecology in respect of its interaction with its environment or symbiotic relationships. Thank you. Next slide.
And I think we might not have Elsa here as well. So we'll go forward and come back at the end if we find Elsa.
Good morning, everyone. My name is Mukesh Bindalkar, basically from India, and currently I'm doing a PhD on eDNA analysis in ST, Spain. So basically, this is my first objective of my PhD project, where I am using eDNA analysis, particularly for finding out the distribution of two significant diatroma species, sea lamprey and European shad. So background is as we know that the climate change continue to the pressing stress on our aquatic ecosystems, and it is not affecting the biodiversity at large, but also pushing several species out of their native range, and it happened to with this species. And for this, we design specific assay for individual species, uh, qPCR assay as well as digital PCR assay. So you can see here, basically we collected the water sample from 4 countries: UK, Ireland, France, and Spain, and almost the network of 46 river basin. And for enhance the interpretability of our eDNA finding, we also compiled the secondary database on the basis of the historic evidence and the literature. And we categorize the species expected to be there in the past, species not expected to be there. So you can see here, the left-hand side is our eDNA analysis, and this the color code is right-hand side is the secondary database. So as you can see here in this region, Spain or in other regions, the sea lamprey, mostly sea lamprey, we were not detected in any location except from this Galicia region in Spain and 2 rivers in UK, and we could not find any traces of eDNA in all other rivers. But interestingly, when we did the same sample in digital PCR along with the addition of the estuarine sample and the ocean sample to see the transit and to quantify the stock population. So we detected, and it's when we reading the paper, like digital PCR outperform the qPCR, and I was very happy when I see this quantitative analysis where our eDNA could not find any sea lamprey. So yes, this is my first objective, and now I'm working on the estuarine species and kind of meta barcoding, and maybe third objective is to be like ocean. So here I'm more interested to Like, is there any short-term training program for 3 or 4 months to learn little bit, means better insight in the water sampling, a collection of eDNA and bioinformatics? So it would be great for me. Thank you very much for this microgrant. It's a lot. Thank you very much. Next.
Okay. Oh. Um, Hi, everybody. My name is Pianan Hanpishanchai, and I'm a researcher at the National Center for Genetic Engineering and Biotechnology in Thailand, Bangkok. And my research is investigating the microbiome in the subway system, especially in the bioaerosols of the subway in Bangkok, Thailand. So we are collecting bioaerosols at the subway station over 12 months during rush hours and non-rush hours. And we are doing that because we think that the microbes and environment in the subway system is very, very important to human health, especially like more and more people in the cities are using subways system for commuting to go some places. And so for this project, we call it Subway eDNA project. We do the 16S and ITS metabarcoding, and also shotgun metagenomic sequencing to get the metagenomic sequence of the microbiome in the subway and analyze those sequence. And so far, we have got some good results so far. And for example, like we found that some bacteria and fungi and some archaea and some virus in our samples that we got from the subway. And for example, the dominant bacteria are proteobacteria, firmicutes, and actinobacteria. And the dominant fungi are Basidiomycota and the Ascomycota. And we found that these bacteria and fungi are distributed differently in different months of the year. So their occurrence may be associated with different seasons and also associated with the many environmental factors such as particulate matters. And also, we also 19 species of the bacteria found in the Bangkok subways were also found to be the dominant bacteria that reported in the paper in Cells by David Danko from Cornell University. So, and also, we found that most of the bacteria and fungi are associated with human sources, especially human skin and human respiratory tract. And the microbes are also linked with sources from outside environment, such as the from soil and wastewater. So this suggests that the microbes that are in the subway system come from both human sources and from outside environment. So, and finally, our people in the lab also start working with eDNA in other environments as well. Such as we are working on eDNA to analyze microbiome in the Mekong River system, and eDNA in the Cass Cave. So I will be happy to collaborate with other people in many aspects of the eDNA in this environment. So, and finally, thank you for listening, and thank you for the eDNA microgrants for giving me this chance to give a talk in this meetup. So thank you. Next slide.
Can you hear me or see me?
Yep.
Okay, thank you. Hello, everyone. My name is Thais Hasselopoulos, and I just has been awarded my PhD recently here in Australia, working with eDNA and with frogs and fungal eDNA. Originally, I'm from Brazil, where I also did my master there, also with eDNA of frogs from rainforest there. So basically, in the last 5 years, I've been sampling water from rainforest here to try to find both the fungal DNA and frog DNA that occur in this area. I don't know if you remember, but back in 2019, Australia had like the worst bushfire they have ever seen recently. Australia is prone to bushfires just like the US and Canada or Europe. But in 2019, we had the worst one. And that was because the Australia was suffering with like 2 years of extreme drought and extreme high temperatures. It was raining less than the last 100 years we have seen. So many of the streams in Australian rainforest was dry, and which was not a good time for frogs to be living. But I don't know if you're aware about this chytrid pathogen, which is a fungus that had spread all over the world and is infecting frogs. But this fungus depends on moist and water to transmit between frogs. So maybe the bushfire and the drought would be a good thing for the frog because the fungus couldn't survive in those conditions. And so I was sampling water to see if we could still detect the pathogen from the streams where the fire was occurring and comparing to areas where the fire didn't occur. And also we've been sampling water since then to see if the things changed since the bushfires were over, and also the water quality have changed, because during the bushfires, the government used a lot of fire deterrent thrown by airplanes to try to stop the fire, which could have affected the water quality and maybe affected the fungus. So yeah, so we're still sampling water to compare and analyze the water quality and use eDNA to detect both frog and fungus DNA that have survived on those forests. And that's it.
Well, I was trying to type questions, and that was very fast. This is what happens with a smaller group, that we ran through the presentations very quickly, and now we get a bit of time to talk to each other, and we don't have to take the whole time, but this is an opportunity to start to create those kinds of communities that we're trying to create. Thais, I was trying to ask in the chat whether you're using qPCR. It seems you know which species you're looking for, so I'm guessing qPCR, is that right?
Yeah, I'm doing qPCR for the fungus because the fungus is just that species and we know which one we're looking for. But for the frog, we actually send for sequencing and try to use metabarcoding approach to do that, which is quite hard. I don't know. I'll be curious to know how other people with other groups of animals with eDNA face this challenge. But for frogs, we don't have many sequences available online. And it was a surprise to get here in Australia and see that basically almost no frog has the sequences available. So we have to I have to go to museums and try to get tissue samples and start from scratch to build this reference for eDNA.
Wow. And I know in Australia, they're building a national reference database, and
Yes,
maybe you benefit from that.
Yeah, but yeah, it started after my PhD.
Not
So I'm still trying to actually get knock on people's doors and ask hi, do you have the sequences for this frog? Because
excellent. And so I think that maybe is a nice place to jump off for a larger conversation about the methods that people are using. We just heard people using quite different. Oh, Eiley has a question. Go for it.
Well, I was going to say before we start a new discussion, Kai has his hand up, so I'd love to hear Kai's question.
I didn't see that. I'm sorry.
I didn't think you saw. Go ahead, Kai.
Why don't we continue this, and then at the end of this thread, I'll come back.
Oh, sure. Just, I guess the thread that I was going down was that where I think we've had good luck if we know what we're looking for, using a single species assay makes sense, a qPCR, ddPCR assay. If you have a question of what's there, we end up using metabarcoding. And it sounds like that's been your experience as well. And I think for many of you, that's the case. Of course, with metabarcoding, you don't get every species that's there because of the idiosyncrasies of the different primers, but it's a broad spectrum of getting many things. So I thought I would pitch that question to the group. Is that consistent with how you see things? Perhaps Joyce, you're a really interesting example where do you know exactly what you're looking for? And you're looking for is the microbiome changing? But is it, I suppose you don't know in which way it might be changing.
Exactly, that's what we're looking for. We don't know what changes are driving this disease, or what changes are in the microbiome during the disease. So we're basically aleatory making it to see what changes we can observe.
Yeah, great. Are there any other, before we go to Kai's, whatever Kai's thread he's going to pull, any other, anybody follow up on that same idea? Do you have a disagreement perhaps, or other ways that you think about using the genetic information that you're getting from the different environments you're sampling? Okay, hearing none, let's go to Kai Lee.
Okay, my question was really for Pianon, if she's still on the call. What caught my attention was that she's working in the urban environment, which is really quite unusual, and I wanted to understand more whether there are decisions that are being made because of the data that she's collecting. Or Pianon, are you studying? Are you kind of doing the natural history of the microbiome of the urban places?
Yes, pretty much. But we also report our findings to the survey authorities in Thailand, and so and they read our reports, and they say that they are quite kind of happy that we don't find any serious human pathogens in the survey system. So and they say that they might report this finding to the higher officials and say that, oh, our system is kind of maybe quite safe for now.
So I wonder, for example, with COVID coming back in many countries, whether you might be, your data might help to understand whether there is a COVID outbreak going on in the subway system and cause people to either to warn riders to stay away or to wear masks? You see what I'm talking about here?
Yes, yes. Yeah, we have thought about that, but unfortunately, our samples are like we only extract the DNA from the sample, and we have not extracted RNA yet from the samples. So that's the good question. So the next step may be like we can also extract both DNA and RNA from the samples, and so we can analyze the COVID-19 virus.
Okay, great. Thank you.
I was going to
Thank
follow
you for
up
your
with a
question.
question. Yeah, I was going to follow up with a question as well, if you don't mind, with the differences
Yes.
between sort of working in a really urban environment versus a more natural environment and water samples or something else, and just ask if there's particular challenges. Like we deal with inhibition a lot in water samples with things like humic acids and things like that. So
Yes.
when do you have particular issues from taking samples in the urban environment? And if so, I don't know how much you know about the specific challenges in non-urban environments, but what's kind of the hardest part of the sample processing pipeline for you?
Yeah. Well, first of all, the first challenge would be like getting permission to do the sampling in the subways or in urban settings, because we need like permission to do that. So we need like MOU and things like that. But like for our work, our main challenge for our work is that so how to get enough DNA from the air. So and we try to use the way to get DNA as much as possible in the shortest time possible. So we kind of I don't have a picture to show you right now, but so we try several like air sampler, like the device that would to collect the air samples, but we finally end up using kind of the ventilation fans, and we modified it so that we can fit at least 27 filters on the fan, so that we can collect the samples with like 27 filters like at a time. If you're like we already published a paper on that in PLOS One.
Yeah, well, I would love if you could put that paper in the chat. That's amazing. 27 filters. And I don't know, again, there's I don't know how much you know about keeping up with the non-urban literature, but we had a visiting scholar who did air filtration from a zoo. And I know it took them a long time to find the right kind of filter for the same issue of accumulating enough DNA. So yeah, if you don't mind putting that in the chat, that would be great. The paper link.
Sure.
Thank you.
And one of the reasons we did that is because like we don't have enough money to buy like the really expensive like air samplers. So we kind of find a way to use the like sampling with like lower cost. So it worked for us. So.
I think this is such a good it's a good example of a challenge that many of us face, where there is the challenge of cost, certainly, but also the challenge of just the amount of DNA that you can get. Right. So that's pulling DNA out of the air, but also Antonio, I imagine, there are limited amounts of DNA. You need a lot of DNA maybe to get a whole mitochondrion, I'm guessing. And same thing with Mukesh is saying, I asked him the question in the chat there, that is digital PCR more sensitive than qPCR? And in general, that's been, I think that's been our experience. I think that's the general word on the street. But so that's one technique for maximizing the benefit of a sample with rare DNAs. If you know what you're looking for, you can use, you know, the
Right.
most sensitive method possible. But I love the idea that you've gone with 27 filters, and you say, well, we're just going to get as much DNA as we possibly can get out of the air.
Yeah.
And
Yeah.
in the case of the marine environment, people very often do that with like a bigger water filter, and they'll try and filter 50 liters instead of 1 liter, and just try and maximize the chance of seeing what you're getting. So that's just, I'm trying to make linkages between these very different projects, but I suspect that we all, you know, have similar challenges like that.
Yeah.
On this subject, can I ask you how long were you filtering the air? Because I was just thinking on people trying to detect the fungal DNA that I studied from the air. Because
Mhm.
you know, when forest in the morning has that fog, and they believe that the fungus can also travel through air during that fog.
Right.
And they were trying
Right.
to like vacuum the air with filter as well to try to detect the eDNA, but they couldn't. But I think it's a matter of like the same issue of trying to get this tiny eDNA from the whole forest air. Now I was wondering how long were you filtering the air to get the most of DNA you could?
So we've done the sampling like for every month, and so for each month, um, we do the sampling like for about 3 days, and for each day, uh, we um, we collect samples um, um, like every hour for 6 hours. So we each so for our sample will be like for 1 hour, 1 hour for 6 sample each days. And so for 1 hour, we'll have, or we collect uh, 27 filters, and then we pull the sample, or we pull the filters together, and we extract the DNA from these 27 filters. So so like at the end we so we uh, like so from 27 filters in 1 hour for each DNA samples.
And then that's a lot of extraction. So you
Yeah.
extract all of them,
Well,
right?
we like the way we do it is that from 27 filters, we kind of use the buffer to wash, try to wash the microbes out of the filter into the supernet, and then concentrate the microbes in the supernet by filtering again. And so like kind of like, and then we got after filtering, so we got like one filter membrane left from like after filtering, and so that and we uh, so then we use the last one filter membrane for a DNA extraction. So we kind of like concentrate first.
Yeah, that's a great idea. Great. Well, I think one thing that we'd love to do is have you talking to each other, which is what has just started happening, which is great. I wonder if anyone else has questions for the group or things you're looking
Yeah.
for. Like Mukesh, for example, said, I'm looking for a training, and now we can have our ears open to think, okay, does such a thing exist, and where could we find that kind of thing? I wonder if Joyce is looking for a PhD program, perhaps we can keep our ears open for that. And how can we help you, and how can you help each other?
I am right now searching for some workshop or something about 16S because I know the Fisalia offered one last year, but I wasn't for my master's for that time. So I'm keeping I'm on the look for that one.
And is that 16S because it's so common for bacteria, and that's the standard thing to look at? And is that's why that's why 16S, or is it the broader question of metabarcoding and sequencing and bioinformatics?
Yeah, we are looking at 16S for the project as well, because that's basically a gene that is easy to evolve, or to say like that, to have ¿cómo se dice? Okay, mutations. Nah, mutations. Sorry, I have.
I think it's the same word in Spanish too. It's
Sí,
it's
so yeah, we're looking at that. So I'm trying to learn more about that, because I have not seen I have not seen workshops or courses about that.
I just dropped one in the chat. There's one, Ryan, I don't know if you've ever done it or known, but we've known some of the instructors at STAMPS is the acronym. And they focus mostly on 16S microbial metabarcoding. It's intense because it's in Massachusetts, it's in person. I can't remember how long it is. But we'll also keep an eye out for some virtual ones too, and other ones that we hear of, Joyce, and we can definitely be sure to send anything your way.
Okay, thank you. Thank you. I didn't know about that one.
And then in my case, now I'm transitioned from my PhD to try to find a postdoc in the same area. And also, I would like also a training too, because it was very hard to find the ways to circumvent the issues of trying to find the frog DNA in the water without having that database ready, and trying to things like from the field, trying to select more water or trying to work with a different primer and all this. And I noticed that you mentioned Karen Goldberg from the US.
Yeah.
I don't know her in person. Do you know her? She's just in on my papers that I read about it.
Yeah, let me I'll look her email up, right, and I'll put it in the chat for you. But yeah, she's great. And so she's certainly a good contact. If she's not, I don't know if she is looking for a postdoc right now, but maybe. And if not, she knows everybody. So that's a good contact.
Okay, thank you.
I can't look it up and facilitate at the same time. Somebody else has to talk.
All right, well, maybe if there aren't any more questions, we can start to wrap up the meeting. That sound good for everyone? All right. So we'll return a couple more slides, not too much more. All right, Eiley.
Thanks, Kate. The other thing that we wanted to really get you guys all, well, we have you all together, is in the Google Doc, there is a section of the document that says upcoming events. And what we were hoping was, though virtual meetups are great, if anyone is planning on going to any conferences, we listed a handful that we're aware of. And if you feel comfortable putting your name next to conferences you might be attending, then you could also meet each other in person if you're traveling, or if it's a virtual or hybrid meeting, you could at least, ask what sessions other people are going to. So in the Google Doc, you can live edit. And then also we just put, 3 or 4 on here, but if you know of other meetings that you're planning on going to or hope to go to, put them in the document as well. And we can put them on our website and try to advertise those as well. It's hard for us to keep track of all of the meetings happening worldwide. And that's another great thing about having micrograntees in 40 countries, is you can tell us where you're planning to go to meet other people. So please use that as well. And I will hand it off to Kara, I believe, for the networking tip of the day.
This is just one of many options, but always a good place to start. Now that you all sort of know each other a little bit and have chatted and seen each other's work, it's not a bad idea to take a few minutes and email somebody that was on today's chat, say hello, and start building your network. A few other things is I think we'll put this into the Google Doc. There is a Slack channel about eDNA that is just getting started, and it's been growing, and it's global. And so I think Eily or Kate can put those into the shared Google Doc. So that's very useful as far as networking goes. And then also our listserv, which I have been tweaking because I know it's not the easiest thing to use. So I've been trying to troubleshoot, and I will be sending some emails around through the listserv. And then you all are welcome to also send emails to everybody. The people on the listserv are just the micrograntees, all of you. So 110 of you, our visiting scholars, and a few people we gave a little bit bigger grants to, but it's just the internal environmental DNA collaborative group. So it's not as widespread as the Slack channel. So if you just want to ask a smaller group, and those are all really great ways to sort of get going with networking and starting to build your personal networks. So, oh, and then one more thing. I just want to make a little plug for the blog. Thank you for, I think a few of you have already submitted things for it, and we're posting them weekly. But if anybody else would like to write a very short little blog post about your science, that would be amazing. You can just check it out on our website and see what people have written, and just to get an idea. But you can write things that are more technical or less technical or shorter or longer, whatever you like. But we love to post and advertise all of your science. So thanks.
Okay, and this is the science tip of the day is something that took me a long time to do, which is we all have to write a lot of code, even if we're not good at it, because you have data that comes at you and you have to be able to handle it. And so we're sort of building these skill sets, and that can be slow. So I think a really good tip is take the time to learn shortcuts for yourself. And one of those, my favorite one probably, is if you need the whole path of a file on your computer, there's a shortcut for that where you copy the path rather than copy the file. And that saves a ton of time when you're writing code and you have to refer to other files. But that's just one example of small things that can make a big difference in your day. And that's nice.